Hello everyone. Today we are discussing about genetic code. What is meant by genetic code? The genetic code refers to the instruction contained in a gene that tells a cell how to make a specific protein. Once the structure of DNA has been described, it was evident that the sequence of amino acid in a polypeptide was specified by the sequence of nucleotide in DNA of a gene. The DNA could serve as a direct physical template for the assembly of a protein. The information stored in the nucleotide sequence was present in some type of genetic code. During the expression of a protein coding gene, information flow from the DNA to mRNA by transcription and to protein by translation. Genetic code. The first model of genetic code was presented by the physicist George Gamow, who proposed that each amino acid in a polypeptide was encoded by three sequential nucleotides. The genetic code is a set of rules by which information encoded in genetic material, DNA or RNA sequence, which is translated into protein by living cells. Genetic code is a set of three-letter combinations of nucleotides called codons. There are 64 codons. 61 codons code for 20 amino acids and 3 codons do not code for any amino acids. These are the 64 codons. Characteristics of genetic code. First one, it is triplet code. Second, commaless. Third, the genetic code is universal. The same code is present in all organisms. Fourth one, non-ambiguous code. One codon cannot specify more than one amino acid. Fifth one, it is non-overlapping. The genetic code is non-overlapping. Each nucleotide along the mRNA would be part of one and only one codon. Sixth one, initiation codon. Once a ribosome attached to the mRNA and begin to read the first codon, it always moves along its conservative blocks of three nucleotides. To ensure that the proper triplets are read, the ribosome attached to the mRNA at a precise site termed the initiation codon AUG, which automatically put the ribosome in the proper reading frame so that it correctly put the message from that point on. Seventh one, termination codon. The three codons that do not have a special function, they are recognized by the ribosome as termination codon, that is UAA, AUG and UGA, and this cause the reading of the message to stop. Eighth one, degenerate code. The amino acid will be specified by more than one codon. A code of this type is said to be degenerate or reductant code. Next one, the code has polarity. The code has definite direction for reading the message. Wobble hypothesis. It was proposed by Francis Crick. The first two bases of a codon must pair correctly with the anticodon according to Watson-Crick base pairing rule, but the last base of the codon can wobble from its normal position to form an unusual base pair with the anticodon. This proposal was called the wobble hypothesis. Crick proposed that a G in an anticodon can pair not only with a C in the third position of a codon but also with a AU. This would give the wobble base pair. One of the unusual nucleotides found in tRNA is inosine, which has structure similar to that of guanosine. This nucleoside can originally pair like G, so we would accept it to pair with C or U in the third position of a codon. But Crick proposed that inosine would still form another kind of wobble pair, that this time with A in the third position of a codon. That means an anticodon with I in the first position can potentially pair with three different codons ending with C, U or A. The wobble phenomenon reduces the number of tRNA required to translate the genetic code. According to the wobble hypothesis, a cell should be get only 31 tRNA to read all the 64 codons. The Wobble pairs are GU and IA. Some organelles have evolved with fewer tRNAs that are required to translate all the sense codon. In this case, codon with U in the Wobble position can apparently translate codons with all the four bases in the last position by super Wobble. Variation in the genetic code. The standard genetic code is a common language used by almost all organisms to translate nucleotide sequence from DNA and RNA into the amino acid sequence of protein. The standard code is not used in all cases, and some organisms and organelles use variant codes. The genetic code is a basic grammar for translating the genetic information into amino acid sequence of protein. Most organisms use the same genetic code, so-called standard genetic code, assuming that all domains of the life evolved from a single organism. The transfer RNA is an adaptive molecule that links the genetic code with its corresponding amino acid via codon-anticodon interaction. A wide variety of chemical modification in the tRNA and codons plays an important role in acquiring protein synthesis by restriction, expansion, and alternation of codon recognition. Natural instance of genetic code variation in several organisms and organelles violated a concept that the genetic code is frozen, but proposed it has continued to evolve. Recoding is an alternative usage of genetic code in some special biological contexts, including programmed ribosome frame shifting, stop codon read-through, insertion of non-standard amino acids, and translational bypassing. That's all about the genetic code. Thank you.